Introduction. Living organisms are in constant need of energy, which is supplied to them by a set of reactions and chemical processes. A group of chemical transformations and reactions which are critical and imperative for sustaining the different activities of daily life of an individual or a living being is known as metabolism. The name metabolism is derived from a Greek word metabolismos. These are reactions which are catalyzed by enzymes and aid various functions like growth and reproduction. It also allows the organisms to respond appropriately to environment and maintain their structures. The gamut of metabolism also engulfs some of the processes like digestion and the transport of substances into and between different cells, which occur regularly in living organisms. In case of such reactions that take place within the periphery of the cell, the processes are termed as intermediary metabolism or intermediate metabolism. The process of metabolism can be classified into two categories, namely catabolism and anabolism. The process of catabolism works on breaking down the organic matter and manifests energy by dint of cellular respiration, and anabolism is the process which utilizes the energy in the making of the parts of the cells such as proteins and nucleic acids. The metabolic system of any organism is the deciding authority in finalizing the nutritious and poisonous substances for the body. The speed of metabolism, the metabolic rate, are the influencing factors in deciding the quantity of food a living being will need to carry on with all the activities of life, and also has a sizable impact on the procedures used in obtaining food. In this chapter, we will understand the metabolism mechanism in some of the macromolecules like carbohydrates, lipids, proteins, nucleotides, etc. Metabolism of carbohydrates. The process of production, storage, and use of carbohydrates in the living organisms is termed as carbohydrate metabolism. In an internal homeostasis, it is majorly the maintenance of the blood sugar levels. The metabolism of carbohydrate is a highly conserved process, and it takes place even within bacteria like the lac operon of E. coli. The basic structure of carbohydrates consists of carbon, hydrogen, and oxygen in the form of following generic formula CnH2nON. However, not all forms of carbohydrate can be metabolized. One of the most used carbohydrates in all living forms is the glucose, which is a monosaccharide. However, larger molecules, mainly disaccharides, oligosaccharides, and polysaccharides are also metabolized within the human bodies. Other examples include fructose and galactose. Carbohydrate metabolism manages many processes within the body. Some of them are cellular respiration to produce ATP involves catabolic reactions that often rely upon the use of sugars. Also, particularly, the brain is dependent a lot on the metabolism of glucose. The main source of carbohydrates is the food that we intake in the form of breads, cereals, pastas, potatoes, etc. After the food intake, the metabolism begins with the gastrointestinal tract, which is the small intestine. It is here that the simple monosaccharides are absorbed through the thin epithelium into the bloodstream. The enzymes called glycoside hydrolases breaks down the larger molecules. Also, the pancreatic enzyme known as amylase is responsible for converting starch into disaccharides and trisaccharides to later be hydrolyzed into glucose. Some of the carbohydrates, mainly polysaccharides, are not digested at this stage and they are subsequently moved to the large intestine. The metabolism of starch, which is alpha glucose, is pretty simple and straightforward. However, beta glucose cannot be digested, and this is the reason why some polysaccharides, such as cellulose, are never digested in humans. Cattle are able to digest cellulose and some other ruminants as well. The monosaccharides levels need to be constantly maintained as they are passing continuously into the bloodstream in large volumes immediately after intake of food. If the regulation of monosaccharides is failed, then it can result in metabolic diseases such as diabetes mellitus. The three main hormones involved in this glucoregulation are epinephrine, glucagon, and insulin. The pancreas produces insulin, which is solely responsible to have the additional digested glucose moved to fat tissue, the liver, and muscles from the bloodstream. It is stored in the liver and muscle as a glycogen in a process called glycogenesis. If the concentration of glucose in the blood drops, then the stored glycogen is used in order to avoid the state of hypoglycemia, clinical low blood sugar. This is done through a process called glycogenolysis, the production of glucose through the breakdown of glycogen, which is prompted by epinephrine and glucagon. For structural support, the carbohydrates are also stored within the cell. The processes are of utmost importance for providing the energy needed by the body at a later point in time. However, at times, the monosaccharides are needed to provide energy on an immediate basis, and for this, a process called glycolysis sets in. The glucose is converted into pyruvate during the process of glycolysis through a series of intermediates in a process which releases energy as ATP. One glucose monosaccharide can produce two molecules of pyruvate. During the formation of 3-carbon sugar phosphates, which are the intermediates, electrons are transferred to the enzyme NAD+, and ATP is produced. While undertaking extensive gymming and exercise routine, pyruvate can be converted back into glucose through gluconeogenesis as it causes us to respire anaerobically. Glucose and oxygen are metabolized to create energy during aerobic respiration, releasing carbon dioxide and water as waste products. For quick and short-term energy requirements of the body, the carbohydrate metabolism is an efficient process. In addition, the large molecules of polysaccharides are also relatively easier to hydrolyze than the fats and proteins that we use for energy. Metabolism of lipids. The primary angle of metabolism of lipids includes the oxidation of fatty acid to generate energy or the process of the synthesis of lipids, which is called lipogenesis. The processes of metabolism of lipids and carbohydrates are closely related to each other as eventually they get converted to fat. Sometimes the rhythm of metabolism of both is disturbed by diabetes mellitus. The hydrolysis of the lipid in the cytoplasm to produce glycerol and fatty acids is the first step in lipid metabolism. Glycerol, being a 3-carbon alcohol, is easily metabolized into glycolysis, dihydroxyacetone phosphate. Also, the reaction is readily reversible if glycerol is needed for the synthesis of a lipid. The hydroxyacetone obtained from glycerol is metabolized into one of two possible compounds. Dihydroxyacetone may be converted into pyruvic acid through the glycolysis train to produce energy. Also, the dihydroxyacetone may also be used in gluconeogenesis to make glucose 6-phosphate for glucose to the blood or glycogen depending upon what is required at that time. With the usage of fatty acid spiral, the fatty acids are oxidized to acetyl CoA in the mitochondria. The acetyl CoA is then finally converted into ATP CO2, and H2O with the help of the electron transport chain and the citric acid cycle. Fatty acids are mainly synthesized from carbohydrates, but there are times when they are synthesized from proteins as well. The carbohydrates and proteins are initially catabolized into acetyl CoA. The acetyl CoA enters the citric acid cycle or is used to synthesize fatty acids in a process known as lipogenesis, depending on the requirements of energy. In the fatty acid spiral, there is a single reaction which directly uses ATP, and that is the first step. A large amount of energy is released and restored as ATP during the oxidation of fatty acids. The ATP is formed from both the fatty acid spiral and the citric acid cycle. The process of conversion of excess carbohydrates and proteins into fatty acids and triglyceride happens in the liver, which are then exported and stored in adipose tissue. The liver synthesizes large quantities of cholesterol and phospholipids. Some of this is collaborated with lipoproteins and made available to the rest of the body. The remainder is excreted in bile as cholesterol or after conversion to bile acids. Metabolism of amino acids. The process of amino acid metabolism is slightly complex as it includes a large number of metabolites which are found in dietary proteins, vitamins, nutritional supplements, and health supplements. The body makes tissue proteins which are further broken down to produce energy, and all this happens through the process of amino acid metabolism. Amino acids are known as the body's building blocks because they provide the fuel needed for growth and healthy functioning of body's tissues and organs. As it is a known fact that protein molecules are made of long chains of amino acids bonded to each other, there are several steps involved in the metabolism of amino acids. Firstly, food proteins are broken down into amino acids, due to which the amino acids are absorbed into the bloodstream. These amino acids are then used in body cells to form new proteins for energy. In the gastrointestinal tract, protein foods are digested to amino acids. The process begins in the stomach and is concluded in the duodenum of the small intestine. The amino acids are then absorbed and are carried in circulating blood to the liver. They are then further distributed to different tissues where they form a series of amino acid pools. As the body's different organs require protein synthesis for energy, they are supplied by these pools of amino acids that are stored in tissues. If the amino acids are in excess, then the requirements of the body, then they are by liver enzymes into keto acids and urea. Keto acids are then used as sources of energy, converted into glucose or stored as fat. Urea is excreted from everyone's body in sweat and urine. The metabolic superachievers in the body are the hepatocytes. They play critical roles in synthesizing molecules that are utilized elsewhere to support homeostasis, in converting molecules of one type to another, and in regulating energy balances. The most important functions of protein metabolism that occur in the liver are deamination and transamination of amino acids, followed by the conversion of the non-nitrogenous part of those molecules to glucose or lipids. Several of the enzymes used in these pathways, for example, alanine and aspartate, aminotransferases, are commonly assayed in serum to assess liver damage. Removal of ammonia from the body by synthesis of urea. Due to the toxic nature of ammonia, they need to be removed quickly and effectively from circulation to avoid any kind of disease which could affect the nervous system. A frequent cause of such hepatic encephalopathy in dogs and cats are malformations of the blood supply to the liver called portosystemic shunts. Synthesis of non-essential amino acids. Hepatocytes are responsible for synthesis of most of the plasma proteins. Albumin, the major plasma protein, is synthesized almost exclusively by the liver. Also, the liver synthesizes many of the clotting factors necessary for blood coagulation. The resting muscle also plays a role in the metabolism of amino acids, and 6 amino acids are metabolized in resting muscle. They are leucine, isoleucine, valine, asparagine, aspartate, and glutamate. The amino groups and the ammonia required for synthesis of glutamine and alanine are provided by the amino acids. They are released in huge amounts in the post-absorptive state and during ingestion of a protein-containing meal. Only leucine and part of the isoleucine molecule can be oxidized in muscle as they are converted into acetyl CoA. The other carbon skeletons are used exclusively for de novo synthesis of TCA cycle intermediates and glutamine. The carbon atoms of the released alanine originate primarily from glycolysis of blood glucose and from muscle glycogen. After consumption of protein-rich food, BCAA, branched amino acid, and glutamate are taken up by muscle and their carbon skeletons are used for de novo synthesis of glutamine. About half of the glutamine released from muscle originates from glutamate taken up from the blood, both after overnight or prolonged starvation and after consumption of a mixed meal. Glutamine produced by muscle is an important fuel and regulator of DNA and RNA synthesis in mucosal cells and immune system cells and fulfills several other important functions in human metabolism. The alanine aminotransferase reaction functions to establish and maintain high concentrations of TCA cycle intermediates in the muscle during the first 10 minutes of exercise. The increase in concentration of TCA cycle intermediates probably is needed to increase the flux of the TCA cycle and meet the increased energy demand of exercise. A gradual increase in the leucine oxidation leads to a carbon drain on the TCA cycle in glycogen depleted muscles and may thus reduce the maximal flux in the TCA cycle and lead to fatigue. The deamination of amino acids and glutamine synthesis present alternative anaplerotic mechanisms in glycogen depleted muscles. One-leg exercise leads to the net breakdown of muscle protein. The liberated amino acids are used for synthesis of TCA cycle intermediates and glutamine. Interactions between the amino acid pool and the TCA cycle are suggested to play a central role in the energy metabolism of the exercising muscle. Another organ that plays a role in the synthesis and inter-organ exchange of several amino acids is the kidney. However, there is very limited information available in this regard. Primarily, the glutamine is metabolized into ammonia by the kidney. This process is sensitive to pH and serves to maintain acid-base homeostasis and to excrete nitrogen. The metabolism of renal glutamine and ammonia is complementary to hepatic urea synthesis. The intestinal glutamine breakdown produces citrulline, which is converted into arginine by the kidney. Renal phenylalanine movement is followed by stoichiometric tyrosine release, and glycine movement is accompanied by serine release. Specific oligopeptides, for example, glutamine dipeptides, are converted by the kidneys to their constituent components before they can be used in metabolic processes. The kidneys are as important as the gut in intermediary metabolism for some of the amino acids. Enriched in glutamine dipeptides is provided by the kidneys, which prove to be disease-fighting needs in many cases. Metabolism of nucleotides. It is a known fact that nucleotides are the information-carrying macromolecules of the cell and are the building blocks of nucleic acids. The two types of nucleic acids are ribonucleic acid called RNA and deoxyribonucleic acid called DNA. Nucleotides consist of three parts. 1. A sugar, either ribose in RNA only or 2. Deoxyribose in DNA only. 2. A nitrogen base, a purine or pyrimidine ring attached to the sugar 1 carbon. 3. A phosphate group or groups usually attached to the sugar 5 carbon. Nucleotides are used to execute biochemical reactions and are important for reasons besides being precursors of nucleic acids. ATP is the most commonly used source, but GTP is used in protein synthesis as well as a few other reactions. UTP is the source of energy for activating glucose, glycogen synthesis, and galactose. CTP is an energy source in lipid metabolism, glycerolipid synthesis. AMP is part of the structure of coenzymes like NAD and coenzyme A. Nucleotides can be synthesized by both de novo, new synthesis from scratch, and salvage pathways, and reuse those we already have. Nucleotides contain one of two kinds of sugar. Deoxyribonucleotides derive the name from the fact that they contain deoxyribose, whereas ribonucleotides contain the sugar ribose. The sugar ribose is a product of the pentose phosphate pathway. Deoxyribose is produced by action of the enzyme ribonucleotide reductase on ribonucleotide diphosphates and is not produced in the cell. There are two kinds of nitrogen-containing bases in nucleic acids, purines and pyrimidines. Purines consist of two fused nitrogen-containing rings with a total of 9 ring atoms. Pyrimidines have only a 6-membered nitrogen-containing ring. Purines and pyrimidines are flat, hydrophobic, aromatic molecules that absorb ultraviolet light 260 nanometers. There are two purines, namely adenine and guanine, which are found in both DNA and RNA, and 3 pyrimidines, namely uracil, thymine, and cytosine, which are important. Salvage of nucleotide bases. The bases used in nucleotides can come both from new synthesis, that is de novo, or from salvage of bases previously made. The figure below highlights the breakdown or synthesis of nucleic acids along with breakdown or synthesis of nucleotides. The bottom nucleobases such as thymine, guanine, etc. and nucleoside monophosphates can be interconverted by action of the enzyme phosphoribosyl transferase on them in the presence of 5-phospho-alpha-D-ribosyl 1 pyrophosphate, that is PRPP. The mononucleotides have a central role in the process. They can be converted to nucleoside triphosphates and DNA, or can be converted to either nucleoside by action of nucleotidases or broken down into nucleobases, that is reversal of phosphoribosyl transferase reaction. De novo synthesis of purine nucleotides. The long metabolic pathways are efficiently synchronized by controlling an early step in the pathway. In purine de novo biosynthesis, the regulated enzyme is PRPP amido transferase, which catalyzes reaction 1 in the figure shown below. Either AMP or GMP alone will slow down the amido transferase, while AMP and GMP together act in synergy. The rate of the reaction can also be controlled by PRPP, phosphoribosyl pyrophosphate. Normal intracellular concentrations of PRPP are below the kilometer of the enzyme for PRPP, so there's a huge scope for increasing the rate of the reaction by increasing the PRPP concentration. Feed-forward activation. Actually, very high concentrations of PRPP overcome the normal nucleotide feedback inhibition. In the bacterium E. coli, purine and pyrimidine biosynthesis are also controlled by genetic regulation. The PURR repressor, when bound with guanine or hypoxanthine, inhibits transcription of several genes in the purine and pyrimidine biosynthesis pathways. Conclusion. Let us now summarize the key learnings of this chapter. It is critical for all living beings to maintain concentrations of glucose in blood within the acceptable limits. Glucose is the main monosaccharide used in carbohydrate metabolism. The maintenance of glucose levels is referred to as glucoregulation. Glucose is stored as glycogen in a process called glycogenesis. Glycogen is converted back into glucose in a process called glycogenolysis. Glycolysis is used to convert glucose into pyruvate, which is used to produce energy. Pyruvate can be converted back into glucose in a process called gluconeogenesis. The liver is extremely active in oxidizing triglycerides to produce energy. The liver breaks down many more fatty acids that the hepatocytes need and exports large quantities of acetoacetate into blood where it can be used and readily metabolized by other tissues. A bulk of the lipoproteins is synthesized in the liver. The process of metabolism of amino acids begins with breaking down of proteins to amino acids in the stomach, which is then absorbed and is carried in circulating blood to the liver. They are then further distributed to different tissues where they form a series of amino acid pools. As the body's different organs require protein synthesis for energy, they are supplied by these pools of amino acids that are stored in tissues. PRPP is the source of the sugar for purine nucleotides. It is synthesized from ribose 5-phosphate. All purine nitrogens come from amino acids, glutamine, aspartate, and glycine. The first step in purine biosynthesis is the conversion of PRPP into 5-phosphoribosyl amine. The amine comes from glutamine and becomes nitrogen 9 of the purine base in the nucleotide.